A very good morning to one and all. Uh, my dear students, today I'm going to talk something about haploid inducers, haploid induction technologies, which are enabling for advanced uh, plant breeding studies. So, in the picture, if you observe, it's a picture taken by Jennifer Johns uh, at the CIMMYT field assistant. You observe two seeds of maize in the hand of the field assistant of CIMMYT. The left one is a diploid one, whereas the right one is a haploid one. So how to are differentiating these two things is because of purple anthocyanin color marker. So let's move on with a fast intro. So basically, you need to understand some terminology before going to this topic, the haploid and double haploids. So what is a haploid? These are plants originating from the gametes that do not go through fertilization process, but can still generate a viable individual. Therefore, haploids contain only one set of chromosomes found after meiosis in male or the female gametes. And this chromosome set, that is corresponding to only half of the chromosome set found in the fertilization product of this zygote or another kind of somatic cells. And depending upon whether a single set of chromosome is derived from a maternal or a paternal side, the plant is referred as a maternal haploid or a paternal haploid. Then what is a doubled haploid plant? This is nothing but the chromosome set of the haploid plant is doubled spontaneously or it is done artificially. So this is necessary since the haploid plants are really frail and have reduced organ size and are not fertile. The most commonly used chemical agent is colchicine for this purpose, that blocks the cell division without blocking the chromosomal duplication. And this treatment just act like a copy paste kind of thing of the haploid genome into the diploid genome. Consequently, the double haploid plants are having all loci are homozygous, and the chromosome creates pure homozygotes and fully inbred lines. Then what are the different methods to produce these haploid plants? So there are numerous methods which can be categorized into two. The first one is in vitro methods and the second one is in situ methods. So in vitro methods have two kinds of things here. It can be either microspore or anther culture or ovary culture or ovule culture. So these are in vitro cultures, and there are in situ methods which include interspecific crosses, pollen treatments like pollen irradiation, or by making use of inducer lines. Then when it is coming to interspecific crosses or irradiated treatment of the pollen, we need to employ embryo rescue technique for the getting of haploid plants, and also by crossing with inducers also we are able to get the haploid plants. But the haploid plants are weak and sterile, as I have earlier told. So these are done uh, stronger or by making a chromosomal doubling using colchicine treatment. Finally, you get what you call the double haploid lines, which are 2n in condition and pure homozygotes. Then introduction of a double haploids. In the last 2 to 3 decades of time, double haploid technology has been emerging as an efficient alternative to the traditional method of inbred line development. The technology essentially samples the segregating gametes of the source germplasm, usually a biparental cross population, or produces completely homozygous lines in one single step. Both in vitro and in vivo methods, as I have discussed earlier, are being used to develop maize DH lines. However, in vivo methods are proven to be more reliable and efficient in large-scale production of double haploid lines, and hence are commonly used in maize. So it was being adopted in many commercial maize plant breeding programs in case of Europe, North America, and China. Although this application of double haploid technology in the tropical breeding programs has been slow, it has now gained a foothold due to intensive efforts of CIMMYT in collaboration of University of Hohenheim of Germany, making the technology accessible both public and private sector organizations. In the recent years, improved maize hybrids have been even developed with the DH lines as a parental lines and released in Africa. Then production of maize lines. What are the different steps involved? There are 4 steps. The first one is induction of haploids. Second, identification of haploids at the seed or seedling stage. Then chromosomal doubling in the haploids, and finally selfing the fertile double haploid plants to produce the seed for DH lines. Then the haploid induction. So both in vitro and in vivo methods can be used to obtain maize haploids. However, in vitro methods have been very little promising to reliably produce large number of double haploid lines required by the maize breeding programs, and hence these are not currently used. In contrast, in vivo haploid induction paved the way for large-scale production of DH lines, and almost all the DH production pipelines in maize breeding programs of multinational companies are now based on in vivo haploid induction. So discovery of the natural occurrence of the haploid maize all around between 1920s and 60s, albeit at a low frequency, very low, something around 0.1%, laid the foundation for in vivo double haploid production. Such spontaneously occurring haploids were used to produce some of the commercial inbred lines of the time and hybrids in USA and Europe during 1950 and 60s. However, the low frequency of naturally occurring haploids coupled with inefficiencies in later production steps have made it difficult to produce DH lines on the large scale. But a major breakthrough has happened with when certain maize genotypes called as haploid inducers were identified, which are having higher frequency of haploid production than compared to normal maize. So this has become a major breakthrough in the double haploid production. So if you observe the picture 1, which is showing the traditional plant breeding programs, wherein you go through a biparental cross, then keep on selfing so that the chromosomal pair differs in the generation 1, and finally at generation 7, something like that, these chromosomes become identical, where finally an inbred homozygous variety of corn is produced with desirable traits. Whereas in case of double haploid corn breeding programs of inbred development, if you observe, we are using an inducer genotype. Pollen is added to the silks of the desired source germplasm, and the resulting kernel is having only female chromosome in the embryo. And then this generation, the mitotic inhibition happens, the chromosomal replication in the cell nucleus happens, the cell division is prevented because of a mitotic inhibitor. Then the chromosomal doubling. Finally, here is your homozygous variety, which is completely homozygous, which can be self-pollinated to produce inbred line. Then classification of the haploid inducers. How many types of haploid inducers are present? Haploid inducers are classified as paternal haploid inducers and maternal haploid inducers based on the genetic constitution of the resulting haploids. So the paternal haploids carry a haploid genome from the male. Paternal haploids carry a haploid genome from the male, whereas maternal haploids carry a haploid genome from the females. Hence their names, paternal and maternal haploids. Coming to paternal haploid induction. So paternal haploid induction, here paternal haploid inducers are used as females, and the source germplasm from which haploids are desired is used as a male. So the paternal haploid induction resulted from mutation of a gene which is indeterminate gametophyte gene IG1. So from the results of mutation, right? After this point, paternal haploid induction has not been commonly used in maize breeding programs because of its a low frequency. How much it is? Just around 1 to 2%. Then the cytoplasmic constitution of the resulting haploids is different from the source germplasm. This is how we shall look. So if you observe the paternal haploid induction, the female is paternal haploid inducer, the male is the source germplasm. The pollen is coming out of this source germplasm, is used for testing the paternal haploid inducer. However, so here the cytoplasmic constitution of the paternal haploid inducer, if you observe the progeny out of the paternal haploid inducer dusted with the source germplasm, has entirely different, has the same cytoplasmic constitution with a differed nuclear genes. So what has happened here? The cytoplasm has been differed in case of cytoplasm. The cytoplasm has been changed. The cytoplasm from A here is changed with the haploid progeny, which has also occurred in the similar case with the rest of the things. Then, however, derivation of the paternal haploids can be particularly useful in conversion of the inbred lines to their isogenic cytoplasmic male sterile forms. You all know much better. Every time cytoplasm is always transferred from the female parent into the progeny. So this is based on that. Then coming to maternal haploid induction, here the female, the source germplasm is used as a female, and haploid inducer is used as a male. So what is the most important advantage is that, as I have told, the female cytoplasm is always inherited to the progeny. So the nuclear genes are also inherited from the female, as well as the cytoplasm is also inherited the female, but these are the haploid lines, the progeny. So this is the most important advantage, and why people got attracted towards maternal haploid induction. So in maternal haploid induction, the source germplasm is used as a female, as I have told, and the haploid inducer as a male. The resulting haploids inherit both the cytoplasm and nuclear genome from the female parent, thereby making them more appealing for the maize breeding programs. The haploid induction based on in vivo maternal haploid inducers. So the picture A here shows a haploid induction nursery where the plants with the tassels are all haploid inducers, and the detasseled plants are from different source germplasm, and the pollination of the ears of the source germplasm with the haploid inducer is shown in the pictures. Then Emerson and Randolph in 1930s have given for the first time have shown that spontaneously occurring haploids in certain crosses are being shown. So the influence of the paternal genotypes on the haploid induction was later elaborated by Chase and led to the search for more pollinator stocks with a high haploid induction frequency, because that's very low. And the first inducer recorded, called as stock 6, produced maternal haploids at a frequency rate of 1 to 3%, which was the highest haploid induction rate, HIR, at that time. And further efforts to improve the HIR and adaptation to different environments led to development of the several stock 6 based inducers with the high HIR. Using the stock 6 derived haploid inducers, several highly effective inducers with a high HIR and good agronomical performance have been developed using during the last 2 decades, and these includes UH400, RWS, MHI, and PHI, which were being developed by University of Ho Hin Hing. And these are all adapted to temperate environmental conditions, and these have laid the foundation for large-scale production of the DH lines in commercial maize breeding programs. And the HIR percentage is somewhere around 6 to 15%. So 6 to 15% of the plants produce their haploids and rest of them are diploids. Always you are getting a mixture of the lines, the haploids and the diploids. And before 2012, haploid inducers with good adaptation and high HIR were not available for tropical environments, especially in case of public domain. Then what is a haploid inducer? These haploid inducers are routinely used in the plant breeding programs of maize, especially in maize only it has been used, thus represent as an exception. The haploid maize inducer lines all derive from a particular genotype discovered in 1950s that possess the ability to induce the development of the haploid embryos on the maize line of interest upon pollination with this inducer pollen. So this is how the haploid inducer line works. As I have told earlier, the pollen from the inducer line triggers the development of egg cell into an embryo containing only a haploid maternal genome, and this process is referred as in vivo gynogenesis. The maternal haploid production is called as in vivo gynogenesis. Then tropical inducer lines. The first generation tropically adapted haploid inducer lines, which are referred as tails. So to enable the public and private sector maize breeding programs in tropical and subtropical ecologies to adapt to DH technology, CIMIT and the University of Hohenheim together developed these tails. This was first accomplished by transferring the maternal haploid induction trait from the temperate haploid inducers developed by UHO, and these inducers exhibited a better agronomical performance than the temperate haploid inducers and have similar haploid induction rates somewhere around 5 to 8% in the tropical conditions. These first generation inducers were distributed to several public and private sector organizations worldwide under specific material transfer agreements. And however, there was a further scope to derive superior tropically adapted haploid inducers with a high haploid induction rate, better plant vigor, better standability, flower synchrony, and with a tropical mesodermplasm, and also tolerance or resistance to foliar diseases and ear rots. Has gone for the second generation of tropically adapted haploid inducers. These are called as SIM2G tails. Recognizing the scope to further improve this first generation tails for various traits, CIMIT initiated the development of the second generation of haploid inducers for the tropics. The selections were made for higher HAR and superior agronomical performance, and the SIM2 tails showed higher HAR as expected, up to 8 to 15%, and under CIMIT tested environments in Mexico and Kenya, besides better agronomical performance in terms of vigor, plant vigor, synchrony with the source populations, and better standability and resistance to foliar diseases and ear rots. The haploid inducer hybrids developed using these SIM2G tails exhibited greater heterosis for the plant vigor and the pollen protection while maintaining similar haploid induction rates. And these are well suited for open pollination with the source populations in isolation nurseries. And would like to share this specific quantity of the seed of SIM2G tails and will grant the authorization for use of these to interested applicants after signing an MTA with the specific terms and conditions. The intellectual property of the SIM2G tails will remain with CIMIT. Then what are the limitations which are associated with maternal haploid induction? There are several reproductive abnormalities like kernel abortion, segregation distortion, heterofertilization, and twin plants. So the kernel abortion is a well experienced phenomena which is associated with the maternal haploid induction. The segregation distortion is that the proportion of haploid and diploid plants that are obtained is always deviating from the expected frequency, that is a segregation distortion. And heterofertilization is nothing but double fertilization is affected by different pollen from different sources. This may be attributed to delay in a little gap between the pollen falling on the surfaces. And also twin plants, this may be because of the splitting of the embryo into two, which may result in a haploid diploid, diploid diploid, and haploid plants types. Then haploid inducer development and maintenance. The main objective in haploid inducer breeding are improving HAR, integrating new markers for haploid identification, and improving the agronomic performance. There may be a need to develop the inducers that suits to target environments, and also HAR can be improved through selection of transcriptive segregants that show higher HAR compared to parents in crosses involving either two inducers or an inducer with a non-inducer. To develop a new haploid inducers, a breeding program has been proposed based on the pedigree method, which involves selection of the individual plants based on highly heritable traits in F2 generation, followed by selection among the families for HAR. Crossing has also been used if the objective is to improve the adaptation or agronomical traits with the necessary sacrificing HAR, without necessarily sacrificing HAR. And the improvement in agronomical performance for many traits relevant to tropics have been demonstrated through selection in both selfing and backcrossing families derived from inducer-non-inducer crosses. And new markers can be integrated into the haploid inducers by crossing the inducers with the germplasm that has genetic markers and selecting for those marker expressions in the early generations and selecting for HAR in the families fixed for those marker expressions. The one important task in inducing the breeding is precise determination of HAR in plants of the families resulting from inducer-inducer process or inducer-non-inducer process. So R1NJ marker integrated in the inducer plant parent is not of suitable use for this purpose. However, recently it was found that the putative inducer candidates with testers the recessive genes for leaf morphology like ligulous and glossy or herbicide resistance are of good use. In such crosses, only haploids will express the recessive phenotype, thus facilitating visual discrimination between a haploid and a diploid. And the LG phenotype has been extensively used in development of the new inducers. However, any tester that facilitates easy discrimination between a haploid and diploid can be of use. And haploid identification, it's the seed expressing anthocyanin color marker of R1NJ, that is called as Navajo phenotype, is widely used for haploid identification. All these currently used haploid inducers worldwide are equipped with R1NJ phenotype, and R1NJ is in combination with other genes involved in anthocyanin biosynthesis pathways such as A1, A2, C2, BZ1, BZ2, and C1 conditions the purple coloration in the alluron layer of the endosperm and also scutellum of the embryo diploids, which is commonly referred as Navajo phenotype. And haploid seed resulting from these induction crosses expresses anthocyanin only in case of endosperm but not in case of embryo, thus facilitating the differentiating from diploids on visual inspection. Recently, the mechanical sorting has also been optimized based on R1NJ marker expression using the multispectral, hyperspectral, and fluorescence imaging techniques to reduce the time and labor involved in identification of these haploids via Navajo phenotype marker. But reliable estimates of this for the false positives have not been currently available. So the picture 1 shows the years from the induction cross showing R1NJ anthocyanin pigmentation expression, and this the image 2 classification of the seed resulting from haploid induction cross into different categories have been shown. If you observe the diploids, these are having pigmentation both in the endosperm as well as embryo, whereas in the haploids it's only seen in case of endosperm. This is how we are going to differentiate with R1NJ anthocyanin marker, the Navajo phenotype. Then what are the molecular players behind the in situ haploid induction in maize? The inducing capacity of the inducer lines have been recently tracked to 4 base pair insertion at the end of the coding sequence of a gene named not like dad NLD, or matrilineal MTL, or Zm phospholipase A1, which is specifically expressed in the male gametes and encodes a patatin-like phospholipase A localized in the plasma membrane of the male germ unit. The predicted truncated protein is not detectable in inducer lines and loses its plasma membrane anchorage in the heterologous system. How this biochemical function relates to inducing capacity is still a puzzle. It's not resolved, and either a structural or a signaling function has been hypothesized, whereas the loss of this is sufficient to trigger the haploid induction. And quantitative trait loci analysis, QTL analysis, demonstrated additional currently unknown players take part in the process and are going to influence the efficiency of haploid induction. Although R1NJ marker is most commonly used, the use of haploid induction suffers from some practical limitations. So the first one is inhibited by the dominant anthocyanin inhibitor gene like C1-1. It has been shown that R1NJ marker expression is inhibited in a significant proportion, up to 30% of the tropical elite breeding lines. So here Navajo phenotype is of no use because it is not going to discriminate between the haploid and the diploid. And however, we need to find more marker types for this discrimination of the haploid and the diploid. Recently, haploid inducer lines have been created in model plants like Arabidopsis thaliana, Brassica junctia, and maize by use of engineered centromeric histone 3 (CH3). However, this haploid induction method has not been reported in plant breeding programs so far. And these are found to be the key players in case of haploid inductions. The University of Hohenheim, CIMMYT, Syngenta, and many more multinational companies have their minds involved in the haploid induction program. Thanks for listening. Thank you.